Is everyone able to see my slide? Yeah, we'll start the class now. Uh, I hope I'm being audible. Good evening. For this week, we will look at a few sample problems based on the lectures for week 9 of NPTEL Genetic Engineering. So the first question is, electrophoresis of a molecule is carried out in a solution in the, and four different options are given here: horizontal gel electrophoresis, moving boundary electrophoresis, vertical gel electrophoresis, and zone electrophoresis. So we can take a couple of minutes, and whoever wants to answer can first try to eliminate the incorrect options like we used to do and justify why a particular option is incorrect. We can take a couple of minutes, and you guys can start unmuting and justify why an answer is incorrect. Okay. Yeah. Does anyone want to answer? Chandrima, Rajata, and Yogita. You can unmute and start with justifying why a particular option is incorrect. Yeah, Chandrima.
Sir, may I tell that incorrect option?
Yeah. Start with one. Okay, we'll just start with what electrophoresis is. From there, we'll then go to one particular incorrect option.
Actually, electrophoresis is a technique through which we are using the electric charges. We are separating the molecules on the basis of the electrical charge. Actually, on and also on the basis of the molecular weight. We are using the electrical charge and we are separating the molecules on the basis of their molecular weight.
Are these two properties linked to a particular property? As in,
Yes, sir.
do you? So here you're saying there are two factors that would govern the movement of molecule. One is charge, one is mass. So are you having a control over any of these properties? Are you trying to normalize or do something such that one of these properties do not really matter?
Mainly, we are focusing on the charge to normalize that.
Okay, so I'll try to rephrase the question that I was asking. So let's say there are different proteins. We use this for majorly biomolecules, right? So let's say
Yes,
there are two different proteins. One is positively charged at a particular pH, the other is negatively charged at a particular pH. Now,
yes.
will you be able to perform electrophoresis in that case?
Uh, we can because somehow at we have to create in that pH if we are creating that media. If it is positively charged, it will go to the uh, cat anode, and the negative ones go to the cathodes. So it will go to move to their respective electrodes.
Hmm, right. But conventionally, is that how we do electrophoresis? What you said is right, and it would depend on the pH of the buffer. Of course, we said that in this particular pH of the buffer, the proteins are positive and negatively charged respectively. But conventionally, is it possible to do it this way?
Uh, Actually, it is not exactly.
Hmm.
Uh, Actually, we are used to work on a particular charge in that case generally. Suppose if we are working on a positive one, so we are working on that, or we can work it on the negative charge, so we are working on that portion only.
What are the kinds
Generally,
of gel electrophoresis that you are aware of, or electrophoresis that you are
we
aware of
are
generally?
aware of the uh, moving boundary electrophoresis,
Right.
zone electrophoresis,
Hmm.
and horizontal gel electrophoresis.
What about vertical gel electrophoresis?
Sir, or I'm not that much very clear about that vertical gel electrophoresis.
Okay,
Actually,
you know about polyacrylamide gels, right? The
yes, sir.
gels on which we run proteins.
Yes, sir.
That is the vertical gel electrophoresis.
Yes.
And horizontal is the one in which we run nucleic acids.
Agarose gel
Yeah,
electrophoresis.
agarose. Now, think of how you run protein gels and how you run nucleic acid gels. There, do you run it from one particular electrode to another, like positive to negative, something of that sort, or negative to positive? Or is it like you run proteins across both ends? How do you actually do it? The reason I'm
Sir,
asking is with by answering these questions, we'll be able to define which of these two factors is inherently important for electrophoresis in general. Hmm,
sir, here actually depends on one the type of electrophoresis one by one.
hmm,
So, like if we talk about that zonal one, they are using a solid supporting media,
hmm,
which is actually creating this is an inert one which is created between the two electrodes,
hmm,
so that there is no the whatever the sample which is separated during electrophoresis, it cannot immobilize or doesn't come together each other. So it is done in the zone electrophoresis.
okay. What I wanted to say was, you said that for this particular case of two proteins having positive and negative charges respectively, that they will migrate to opposite directions. So conventionally, we don't do it that way, but rather what we do is we normalize the charges on the protein by adding SDS. Do you remember the role of SDS? What is SDS?
Yeah, giving the negative charge and denaturing it.
Yeah, so SDS would denature all the proteins. So in this case, we normalize for the charge of the protein, and the separation is only based on mass. They have, or the proteins have, a specific number of SDS molecules based on the length of the protein, based on the number of amino acids. But the movement is only due to the mass. Okay,
Yes, sir.
right. Yeah, Chandrima was okay. So you were saying that option A is incorrect, is it? Horizontal electrophores. That's what you were mentioning, is it? Or
Sir, actually, I was telling about the incorrect one is like option D.
zone
That is zone.
electrophoresis, right?
Actually, that media is solidified media they are using, sort of inert.
Huh. So
That is why it's
do you think vertical gel electrophoresis and horizontal gel electrophoresis also fall under zone electrophoresis because you're
yes,
telling there is a
it is
vertical media?
a type of.
It's a type, right?
It's a type of zone electrophoresis.
Right. Hmm. Good. What about Rajata and Yogita?
So Rashi told both option A and both option C are incorrect
Hmm. And
because
Renu?
it involves gel.
Right. And gel is a solid media, so you would say here it is not carried out in solution. Okay. And so you all agree that the option would be moving boundary electrophoresis. So this is a particular setup where instead of having a solidified media, rather you would have your solution, your buffer, and your solution here. And these are connected and an electric current goes from the one electrode to another electrode. Now what do you have to maintain here? So when you supply current in a solution, the solution itself gets heated up, meaning there are effects of convection which would move the molecules. But you would want to move the molecules based on the properties of the molecules, which is the mass and the charge. So what you would do is you would take a water bath at a specific temperature like 4 degrees and keep it here so that the effects of the current that give rise to additional heat is nullified. Then over time, movement of ions or molecules happens. And here they use it majorly to check the homogeneity of the sample. I hope that was clear. If not, you can ask me. Okay, the next question is an initiator for acrylamide polymerization is, and four different options are given here. So the first option is Tamid, option B is APS, option C is STS, and option D is bisacrylamide. So we can take a couple of minutes and think about the role of each of these and which of these initiates polymerization. So this is like a general mechanism. Each of you can think about how polymerization would begin and what you need for polymerization. And then try to think about the answer. Okay? We'll take a couple of minutes and then we'll continue again. Okay. So to start with, we'll discuss by what polymerization is. Can anyone unmute and tell me what polymerization is? Yogita? Okay, I don't know if she's there. Can anyone else unmute and tell me what polymerization is?
The polymerization is a process in which we are joining the monomers with particular bonds
Hmm.
to make a polymer.
Okay. Are these bonds covalent or non-covalent?
These are covalent.
Covalent. Right. So, what do you think will be needed for initiating polymerization? Do you know there are different mechanisms to initiate polymerization?
Sir, I'm not aware of that.
Okay, so in chemistry, we might have learned that there are different ways to initiate polymerization. Um, One such mechanism is free radical based polymerization initiation. Um, and here we see that four different components are given: TEMAD, APS, SDS, and bisacrylamide. Which of these options do you think will not be the initiator for acrylamide polymerization?
First is like SDS because
SDS.
SDS job is not that.
Hmm. Do we add SDS to our gels or just the dye?
We use SDS, but in our not in our gel. It is generally we are loading into the sample.
Okay, so actually in labs we use SDS while we are making gels as well. We make a 10% solution and take a stock of 10% solution and add a particular quantity depending on the volume of the gel we are casting. So it is present in the gel as well. Apart from being present in the dye, it is also present in the gel. Okay. So you're saying doesn't play this role. What about other options? Rajata, Yogita, and Appalanaidu?
Sir, option A.
Timid. Okay.
Sir, it's a catalyst, not an initiator.
Okay. So what do you think exactly timid does? What is it a catalyst for?
Decomposition of APS.
It catalyzes the decomposition of APS. Okay. Why does APS have to decompose?
I'm not sure, sir.
Okay. So what is APS, Rajata? And what is timid? Can you give the full forms if you're aware of it?
Timid is tetramethyl ethyl in ethylene diamine,
Hmm. And APS?
ammonium persulfate.
Ammonium persulfate. So you were talking about timid and APS. Now, what you can see is, yeah, who is it? Yeah, Chandrima.
Sir, a initiator and a catalyst major difference is catalyst is a very it's not that much specific. It can work in a chemical reaction, but an initiator is only working when any monomer is converted to a polymer, when we are polymerizing.
But is the initiator a part of the polymerization process? Is it a part of the polymer? We say that we have monomers. There is one monomer unit, there is another monomer unit. Both the monomer units are interacting, they are forming a dimer, and multiple such dimers are forming, and they're forming polymers. Now, is your initiator chemically bound to any of these polymers?
Yes,
Is it?
yes, because enzyme doesn't, but in on initiator does.
Initiator does, or does initiator provide something that would allow you to form the bonds?
Actually, they provide something which is helping to creating that bond.
Right. So they do not form the bond, but rather they provide something which might be some radical, which is what we I was earlier referring to as the free radical method of polymerization. So what we can look from here is that okay, so the options that we've eliminated so far are STS and TEMID. And what other options do you think we can eliminate? APS, bisacrylamide, which of these is incorrect? Is it bisacrylamide or is it APS?
Sir, it will be bisacrylamiide because it is doing a cross linking, not it is a initiator. It works as an initiator.
Right. So can we say it this way? There's an acrylamide, 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 and bisacrylamide is between multiple acrylamides. Is that what you meant by cross-linking here?
Yes, sir.
Right. Good. So what we need to know is ammonium persulfate has this persulfate ion, which is S2O8 2 minus. Now what would happen is TEMED would break down to form a bisulfate radical and a TEMED radical. And this would initiate this APS, which had decomposed, like how Rajata was mentioning, would form this radical that would initiate to form the polymer of acrylamide. That is why this APS is called the initiator and TEMED is not. Shown here is an example where other radicals could play a role in initiation in a different context. But in our case, when you add TEMED and APS, the bisulfate radical is the one that, sorry, the persulfate radical is the one that will uh, give you electrons or give you radicals with electrons rather to form these acrylamide polymers. The next question is the progress of a protein sample in a polyacrylamide gel can be monitored by option A SDS, option B CBB 250, option C bromophenol blue, and option D phenol red. We can take a couple of minutes and then start with eliminating the wrong options. Yeah, we'll start by eliminating. Does anyone want to justify why an option would be incorrect? Yeah, you can unmute. Uh, yeah,
Sir,
yes, Chandrima.
option D, phenol red.
Option B, phenol red. Okay, what is phenol red?
Phenol red is an pH indicator we
Right,
use in the DMEM media that to indicate the pH.
pH change if there is any contamination. Right,
Yes.
right, phenol red. Yeah, Rajata.
Sir, option B,
CBB 250. What is that?
Kumasi Brilliant Blue.
Okay. What does it do? What is it?
Sir, it's a dye used to stain proteins after electrophoresis.
Hmm. Right. But okay, do you know where CBB 250 binds to in a protein?
Sir, I just know it's a post-run stain.
Huh. So it's a dye. It's a staining solution. That is right. It's not a dye, sorry. It's a staining solution, which means it will be able to stain proteins. So what do you think will be the requisite for some staining solution to stain proteins? What property do you think people would have utilized to design these molecules?
Sir, it should be migrating fast.
No. So you told me this is a post-run staining solution, right? So there is no issue with migration. Once your proteins have resolved, you just put it on the staining solution and you see it. So there is nothing to do with migration. What other property do you think people would have used? That is common to all proteins, almost all proteins in nature. 1 minute, Chandrima. We'll listen to Rajata and then we'll come to you. Okay.
Sir, I didn't quite get your question. I am not able to understand if you're talking about the answer or the option which I told.
The option that you were telling. So you told CVV 250 is a post-run staining solution, right? But you told it's a post-run staining solution, which means that it has the ability to bind to proteins, and we will be able to visualize that. Which means if you put it into your protein sample, and if your protein sample is being run, ideally you should be able to see your protein on the gel and the progress. But you said it is incorrect. So I'm asking you first, do you know where the CBB 250 binds to in a protein? That's the first question. Yeah, Chandrima.
Sir, in generally the CBB means for binding to a protein, we require mainly two things. One is a charge.
Hmm.
Another one is we can say hydrophobic interaction
Okay.
because the staining actually is binding with the hydrophobic pockets present in the protein.
Uh, Okay, go ahead. There is something incorrect. We'll discuss that. Yeah.
Actually, first, what we are telling, like the non-polar part, because a protein gen is not a polar, it's not polar fully. So that if there is any non-polar part of uh, the CBB, the Comazi Blenheim blue chain, so
Hmm.
it gets bind to it, that to the hydrophobic pocket, which is creating an hydrophobic interaction between them, or it can work in we can say on the basis of charge.
Okay. And you have two kinds of charges. One is positive, one is negative. So, what do you think CBV does?
CV is slightly positive charged
If it is positively charged, then what are the kind of residues it should bind to?
to the negative as negative residues which is negatively charged.
Okay. Good. You're thinking in the right direction. There are some issues. I'll just point that out. So, you were talking about nonpolar interactions and hydrophobic part, right? Now, think of how you run proteins in a gel. The proteins are denatured, so they are just linearized. Do you think the nonpolar interactions or the hydrophobic regions would be intact once they are denatured?
If they are denatured at that time, it will not work. That pockets is also mean not working.
Huh. So, if they are denatured, it means they are linearized, meaning the pockets do not exist anymore. And in those cases, it shouldn't work. So, CBB was designed to be able to bind to lysine or arginine, basically positive residues. Uh, Although the way in which you thought was right, the mechanism is wrong. But no issues. Good that you're thinking in the right direction. Right. So that leaves us with SDS and bromophenol as the options. SDS we know is a denaturant.
So, one doubt.
Yeah, Rajata.
Will it not bind to aromatic amino acids?
Homasi Brilliant Blue?
Yes, sir.
No, it wouldn't. It is specific for lysine and arginine residues.
Okay, sir.
Right. So, uh, we don't add Comasi Brilliant Blue to our loading die. Rather, we add it after we finish running the gel, like Rajata was mentioning. And the dye that we add is bromophenol blue. And why should we add only this particular dye? Why can't we add any other blue colored dye? Does anyone have an answer for that? What is so specific about bromophenol blue?
Sir, sir,
Yeah, Chandra.
actually in if the DNA is going to from the negatively charged
Hmm.
on the base, actually the bromophenol blue is somehow working that it is also a negatively charged molecule which is
Right.
also going with the DNA. That is why it helps it to tracking, somehow tracking the job. What is the job of the bromophenol blue is that is helping. It goes with them as it is slightly negative charge. On the basis of the charge, it is moving, so we can track that.
So you are saying, I'll just rephrase what you said. First, it is negatively charged, and the molecules that we use are also negatively charged. We run them from the negative electrode to the positive electrode. So the direction of movement will be the same for both the dye and our biomolecule of interest, which is either the protein or the DNA. Second, the way or the speed at which these molecules, these dye or the bromophenol blue in this case, moves is comparable to that of the movement of our biomolecule of interest, which is the DNA or the protein. Now, because these are comparable, we will know where our protein is by looking at the dye. That is what you called as tracking dye, right? Yeah. So
Yes,
these
yes.
are the reasons. Good. You're right about that. So that's why we use bromophenol blue. The next question is which of the following components imparts a negative charge to the protein? Option A, glycine. Option B, SDS. Option C, TEMED. Option D, it's tris-HCl. So here, does anyone know the role of glycine? We have discussed briefly about the role of SDS and TEMED in today's class as well. Tris-HCl is the buffer. But what does glycine do? Glycine is also a part of your uh, electrophoresis system. Does anyone recall what glycine would do?
It's a part of an running buffer.
Right. So it's a part of your running buffer. What would happen if you do not add glycine? What is so special about glycine as an amino acid? Does anyone know that?
In glycine, it's a very good Zwitter ion charge. It's a
N-
good having.
No, what do you mean by very good Zwitter ion? I didn't get that.
So glycine can be used as a buffering agent.
Uh, how would that work? Okay, we'll first look at the structure of glycine. Uh, Based on that, we'll know why it has a particular usage. Does anyone know the structure of glycine or any amino acid in general? So, glycine is the simplest amino acid that one can think of. When you think of amino acids, they have an alpha carbon atom, acid group here, which is in your an amine group, NH2, a H, and an R group, which confers specific properties to your different amino acids. Now, glycine has a hydrogen here, so its R group is doesn't contribute to any charge. Now, Chandrima was mentioning about a good Zwitter ion. So what I probably think she meant is because the R group is not realistically involved in the different isoelectric points, or how the hydrogenation or protonation here or the deprotonation here would be affected by this. So it is simple to study the kinetics. That's what I think Chandrima meant by a good Zwitter ion. So the role of glycine with respect to uh, electrophoresis is that, sorry, this is a wrong slide. So initially in the stacking gel, glycine at the pH 6.8 has a mobility between the one of our proteins and chloride ion, because of which it would sandwich the protein. But once the pH changes to 8.8, this glycine would move forward, having a better mobility, and this is because of the zwitterionic configuration that glycine could have. So that is what I wanted to specify in this particular slide. And Rajata, you were mentioning it has the buffering capacity. What did you mean by that? Can you explain that?
So it helps in maintaining stable pH levels in solutions.
How would you do that? Glycine wouldn't do that, right? Why should glycine do that? Why should
Is
glycine be able to?
Zwitterionics.
It has a zwitterionic configuration, which means that at a particular pH, it would have a particular protonation state. That is
Um,
what it would be. But how do you think it will buffer and adjust the pH?
Sir, it has a pK around 9.6
Okay,
uh, for its amino group and 2.3 for its carboxyl group.
okay.
So it makes this range is effective for buffering in psychological pH conditions.
No, but it doesn't have the capacity to buffer, right? So in order to be a buffer, it has to ensure that pH doesn't change, which means that if you add more of protons, it should take up more of protons. And if you reduce the number of protons, it should release as many protons so that the pH doesn't change. That is what buffering would mean. But here, if you look at the structure of this glycine, it cannot realistically give you more protons or take up more protons, which is why buffers contain a weak acid and a salt of a strong base. So there you would be able to compensate for loss of or gain of protons. But amino acids do not have that capacity, which is why they are not buffering agents. Okay.
Okay, sir.
Okay. Yeah. So this is just a diagrammatic way to show how it moves, but it's not very helpful for this particular slide. The next question, in page, a layer of organic solvent is introduced on top of the resolving gel. Two, four different options are given here. Facilitate entry of proteins, increase production of free radicals, decrease the rate of acrylamide production, prevent the entry of oxygen. Huh. So we will take a couple of minutes and then we will try to identify the right answer, or if you are not sure about the right answer, we'll go with our method where we eliminate the wrong answers and justify them. We'll take a couple of minutes and then continue. Yeah, does anyone want to start with eliminating an incorrect option? Okay, if not, we'll start this way. So, what does a page look like? How many gels are there in a page? How many layers are there in a page?
So there are two.
Two layers. So, what is the top layer and what is the bottom layer?
Sir, upper layer is the stacking gel and lower one is the resolving gel.
Right, good. So, when we are casting gels in lab, if the upper layer is a stacking gel and the bottom layer is the resolving gel, which gel has to be made first? You're on mute, Chandrima. So, there are
Sir,
two
first
layers.
we
Huh,
make the resolving gel.
right. You first cast the resolving gel. And then you cast a stacking gel. Now, do we add organic solvent? If so, what organic solvent do we add?
We can add butanol or isopropanol. It
Isopropyl.
can be.
Right, right. Why do
Sir,
we
why
add?
not ethanol?
Why not?
Why not ethanol?
You can add ethanol. It wouldn't really affect as long as. Okay, there are a few things you need to consider. First, your gel is getting solidified, right? So whatever solution that you add shouldn't enter your gel. That is your first criteria. The second thing is it should stay on top and prevent the gel from interacting with air. These are the two criteria. In lab, we use isopropanol and ethanol also because it's just a very thin layer. It wouldn't really affect. And if you look at the structures of isopropanol and ethanol, isopropanol is heavier than ethanol, which would mean that it could, if isopropanol is able to stay afloat on top of gels, ethanol would also be able to. But people conventionally use isopropanol. That's the reason, Rajita.
Yes, sir.
Okay. So now, why do you think we should add this layer of isopropanol? I was telling that we want to prevent exposure to atmospheric air. But why would we want to prevent exposure to atmospheric air?
Somehow that atmospheric air is interfering in the um, actually we are talking about radicals in the polymerization, so oxygen is somehow interfering in that polymerization by creating a radical, or it is reacting with the free radicals, which could interfere in that polymerization.
Right. That's the right answer. So when you expose your gel to air, the atmospheric oxygen, if it enters, that would prevent polymerization. So it would decrease the rate of acrylamide polymerization, like how Chandrima had mentioned. That's the reason. Right. Good. The next question is the pore size in an acrylamide gel can be controlled by manipulating the ratio of four options are given here: acrylamide timid, acrylamide bisacrylamide, bisacrylamide APS, bisacrylamide SDS. So, does anyone know how the pore size is influenced by factors in the gel components?
Sir,
Hmm.
in this case, actually the pore size is depending on the different concentration of the acrylamide and the bisacrylamide.
Okay. So, how do you think we make the acrylamide solution? Do we have a specific ratio of acrylamide bisacrylamide?
It should be, because it is actually the cross linking, what is happening should be a particular ratio
Hmm.
to create that mesh like structure that was given in the lecture.
Right.
I'm not fully aware about that ratio, but it should be in a particular fashion that to create that mesh like 3-1 mesh like structure.
Right. So this ratio would depend on the kind of biomolecule that you would want to run. The ones that we use to run for RNA gels are different from the ones that we use for protein gels. We usually use a ratio of 29 is to 1 of acrylamide is to bisacrylamide. So in that ratio, we make stocks of our acrylamide solution. Then, depending on the molecular expected or theoretical molecular weight of the solution of the biomolecule, we would decide and cast a percentage of gel. So that way the pore size is being regulated by the acrylamide and the bisacrylamide and not the other components. Yeah, and so the next question is, which of the following component in the loading die increases the density of the protein sample and facilitates its loading into the wells? Four options are given here: glycerol, beta-mercaptoethanol, SDS, and bromophenol blue. So we have discussed this concept earlier, where we know that beta-mercaptoethanol is a reducing agent. It breaks down disulfide linkages. SDS is sodium dodecyl sulfide, which is a detergent, anionic detergent. It gives negative charge evenly to all to the entire stretch of proteins, entire stretch of amino acids of the protein. And bromophenol blue is your loading dye. So even for bromophenol blue to enter the well, one would require glycerol. So glycerol here is the one that is helping increasing the density of both the protein sample and the tracking dye, which is bromophenol blue. The next question is, the pH of the stacking gel is 6.8, resulting in protein being sandwiched between, and four different options are given here: tris and glycerol, tris and bromophenol blue, tris and glycine, tris and SDS. So we'll take a couple of minutes and then we'll start with what is the concept behind this stacking. Okay. Okay. So we were just discussing about the concept of stacking earlier. Now, why is the pH of the stacking gel lower than the pH of running gel? Does anyone know the reason? Okay, so the pH is important because, like Rajata was mentioning earlier, the ions that we use here, which is glycine, has a zwitterionic nature, meaning depending on the pH, it would take different forms. Uh, Now, at pH 6.8, it would take a particular form which has a different mobility. By form, what I'm trying to mean is the amount of charges, the protonation levels, at least at the amino acid level. So, at that particular protonation level, it would be at the exterior end, at the topmost end, having the lowest electrophoretic mobility. So it will sandwich the protein, which is shown as red color spikes here, with chlorine in front. But later, as the pH increases, the charges on glycine would change, which would make glycine come front. So now there is nothing to sandwich the protein and the proteins would resolve. So the reason the stacking is done is so that proteins, when we take it out using the pipette, are suspended in a solution, meaning they are present in a particular vertical suspension. And different proteins could be present at different regions of this vertical suspension. That should not interfere with the resolution of the gel, which is why we try to compress all the proteins to one layer, which is what we do in the stacking gel. And once they come to the resolving gel, they will move only based on their molecular weight. Okay. Okay. So next is an interesting question. Which of the following can be used to monitor the folding states of a protein? Four different options are given here: native page, STS page, gradient urea page, and agarose gel electrophoresis. We'll take a couple of minutes and then try to uh, do this interactively because this is an interesting question. Yeah, does anyone want to try to answer this question? Try answering this question.
Sir, we have to tell the wrong option.
You can, yeah. So based on the discussions in today's class, also we know that native page cannot be used to monitor folding states, but rather oligomeric status. SDS page to get the molecular weight of the monomer. Agarose gel electrophoresis, we don't use it for proteins, rather we use it for
D.
nucleic acids. So that leaves us with gradient urea page. So does anyone know about gradient urea page? Do you know about it, Chandrima?
So in gradient urea is similar to page like the gel is polyacromide gel,
Hmm.
and at different level lanes they are giving the concentration of urea of 0 to 8 molar
Hmm.
to study the protein folding unfolding that due when we add urea to the protein, it is actually urea is creating hydrogen bonds with that protein, which is helping to unfold that proteins.
Hmm. Hmm.
So at various stages, it is various concentration it has been checked, and almost generally in case of proteins, for the 8 molar urea, that protein is completely unfolded and it will be a linear structure.
Hmm. Right. So how do you think in gradient urea page we will get an idea about unfolding? What you told is right. At 8 molar urea, any protein will be unfolded. But how will gradient urea page help in this monitoring the folding states?
Sir,
So,
as well in our lecture, it was like told that it is increasing that hydrodynamic volume. But what is that? I can't understand at that time. When we are
okay,
using that urea.
okay, no issues. We'll look at it now. So there is this website which is I have linked that to the PPT here. I have taken this reference from there. So this is a method where you have different concentrations of urea as across the gel. So as your protein runs, it will come across different concentrations of urea and it would start to unfold at a particular concentration depending on the nature of the protein. Okay. So what they say is single sample of protein is applied across the top of the gel and electrophorused in a direction perpendicular to the urea gradient. As the protein is electrophorused, molecules at different positions across the gel are exposed to different urea concentrations. At positions where urea concentration is high enough to promote unfolding, mobility of the protein decreases because of greater hydrodynamic volume. We'll come to this, Chandrama, of the unloaded form. When the gel is stained or otherwise visualized, the electrophoretic pattern can be interpreted directly as an unfolding curve as shown below for the two forms of the bacteriophage T4 lysozyme. The upper band in the gel contains a mutant form of lysozyme with alanine 160 replaced with threonine, and the discontinuous lower band contains a wild type protein. The wild type protein was applied to the gel first, electrophoresed for 10 minutes, and then the mutant protein was applied and electrophoresis was continued for an additional 20 minutes. The gel was stained with Kumasi blue. The gel reveals two important differences in the unfolding transitions of the wild type and the mutant proteins. The mutant protein unfolds at a lower concentration of urea than wild type protein. The interconversion between the native and the unfolded forms of the native protein is relatively slow, giving rise to discontinuity, while that for the mutant protein is quite fast, giving rise to a smooth protein band. So what they want us to understand with respect to urea phase is, first, the gradient is in one axis. This is the x-axis, and the electrophoresis happens in the y-axis, like we usually do. And you apply protein here. You put your protein here. So now the protein here has a particular concentration of urea. The protein here has a particular concentration of urea. In this particular case, the proteins in this region will have lower concentration, and the proteins in this region will have higher concentration. Now, in the absence of urea, the proteins would migrate as much as they can. But in the presence of urea, like we have discussed in previous lectures and how Chandrima was able to recall it today, the structure of urea is NH2CO NH2, meaning it has the carboxyl groups. I mean, it has the C double bond O carbonyl group, and it has two amine groups, which means that it can form hydrogen bonds with amino acids and collapse their 3D structure. So as you increase the concentration of urea, the proteins in this particular region, they would start to unfold. The folded form can be assumed as the compact form, whereas the unfolded form is the linear form. Now, uh, the volume of this particular wire in this compact form is the 3D form. Here, the hydrodynamic volume is lower. But rather, if you extend it like this, the hydrodynamic volume increases. This is what they had mentioned as the hydrodynamic volume. When the hydrodynamic volume increases, it cannot enter the pore size of the pore of a particular size. But rather, protein molecules like this, with this hydrodynamic volume, can enter. That is why we see that proteins at lower concentrations of urea have migrated to a farther distance than proteins at higher concentrations of urea. And typically, this can be understood as how different protein molecules behave at different concentrations of urea. Was it clear till this point?
Yes, sir.
Okay. And one more thing that they wanted to mention was the rate at which unfolding is happening, which is, now you have this enzyme. Let's say this is the mutant form. Now, the rate at which unfolding happens for the mutant form is very quick, which is why you see a continuity here. But rather for the wild type, it happens slower than the rate at which the molecules are moving, which is why you have two bands. Some fractions are here, some fractions are here, and there is no continuity. Was it clear?
Yes, sir.
Oh, cool. The next question is, vertical streaking of protein bands appear usually due to four different options are given here: partial oxidation of the protein, degradation of the protein, vertical low running current, overloading of the protein. Yeah, we can take a couple of minutes and then start with eliminating the wrong option. Yeah, does anyone want to start with eliminating the incorrect options?
So, option B.
Degradation of protein. Okay, if a protein degrades, what would it look like, Rajata?
Sir, multiple smaller bands.
Okay, there will be bands. It would be this way. Let me draw, and you tell me whether it would look this way. This way, is it? You are running the gel from here to here. Is this how you think it would look like?
Sir, I'm not sure, but it will be multiple smaller bands since it's degraded. Or yeah,
So what you say is the molecular weight will get reduced. So one band after the other, we would see multiple smaller bands.
yes, sir.
Right. Good. So yeah, degradation of protein does not give you vertical streaking. Okay. What else? Would the current really matter?
Sir, it would lead to poor separation of bands.
Huh. So the resolution would be bad. So the molecules will be closer. Is that what you mean by poor separation? What molecule should look like this would rather look like this. Is this what you mean by poor separation?
There will be no distinct separation between them. It will be all joint.
Right, right. So you have eliminated options B and C. Okay. What about oxidation and overloading? What would happen if a protein is getting partially oxidized? Any idea about a particular functional group that you think, if upon oxidation something would happen?
Sir, it might affect its stability.
Huh? Let's. Okay, so you have multiple proteins, let's say like these. And if there is oxidation of these proteins, possibly do you think something might happen? Or in a solution, proteins are getting oxidized. What do you think will happen? In a solution of proteins containing three different proteins, these are oxidized, undergoing oxidation. Which functional group do you think will get the most affected? Or which amino acid?
Sulfur containing.
Sulfur or sulfhydryl group?
Sulfide group. Sorry.
Right. Okay. It's called the sulfhydryl group because it has an S and a H.
Yes, sir, yes, sir.
S and H, which is why we use reducing agent so that it remains as SH. If it's oxidized, it would form random disulfide linkages, and that would lead to incorrect conclusions. So you're coming up with the answer overloading of protein. Okay, I just have a representative example to show you what vertical sticking of protein bands look like. Let me. hope you're able to see. So this is the vertical streaking. This is how it looks like. And that's only because you loaded more amount than for that particular dye to be detected. The next question is a similar question to what we saw earlier in a couple of weeks ago, maybe. The molecular weight of a protein in native and STS pages 240 and 60 kilodalton respectively. Considering that the protein is not a heteromer, the oligomeric status of the protein is option A dimer, option B trimer, option C tetramer, and option D hexamer. So to be able to answer this question, we need to know the molecular weight in the native page form, which is 240, and STS page form. So in the native page, if there are any disulfide linkages, that would be, or if there are any non-covalent interactions that holds up the oligomer, that would be maintained, which is why you see a higher molecular weight in the native page. Whereas in the STS page, these non-covalent interactions that would hold the uh, individual monomers of the oligomer, those will be broken down. So your oligomer is no longer an oligomer, it's just monomer. So the monomeric molecular weight is 60 kilodalton and the oligomeric molecular weight is 240 kilodalton. With that, we say it's a tetramer. That's why it's option C, tetramer. The next question is the electrophoretic mobility of a protein in urea page decreases due to four different options are given here: decrease in charge, decrease in hydrodynamic volume, increase in charge, and decrease in hydrodynamic volume. So here, what one can consider is like we earlier saw with the case of wire, at lower concentrations of urea, your protein is not fully denatured, which means it is still compact, it can still enter the pore. But at higher concentrations of urea, it gets denatured, it's not compact anymore, and it gets unfolded. Now, because it is unfolded, the volume is now more than what it was with the compact form. So, the hydrodynamic volume is increasing. So, the right option here is increase in hydrodynamic volume. The next question is periodic acid shift reagent, specifically stain, and four different options are given here: phosphoprotein, glycoprotein, basic protein, and acidic protein. We can take a couple of minutes and then start with uh, eliminating the wrong options, leading to the right option. Okay. Yeah. Does anyone know the chemistry behind how periodic shift reagent works?
The periodic shift reagent is actually a composition of the periodic acid and Schiff's base.
Uh-huh.
A periodic acid is a it's such an acid which is whose in the iodine it is having that highest oxidation state of
Right.
iodine. So it is
Right.
very highly oxidizing agent.
Right.
Somehow it relates with whichever the molecule it is going through, it oxidizes it.
Hmm.
Somehow
So, huh.
that ship's base is somehow it is working in that staining part because it's a sort of what ship's base is a composition of aniline based and a sodium salt.
Right. It has a fuchsin stain and it has the sodium salt. Yeah, that is right. And you're right about the oxidation as well. Yeah.
It is linking with if when it is oxidized that presence of somehow the group it gets attached that composition of the ship's base it creates some product which is binding.
Okay. Does this happen to all proteins? Or do you know where exactly this oxidation happens?
Actually the periodic acid actually works with the carbohydrate which actually glycol which
It's
is oxidizing to aldehyde.
so is the one that you're talking about is the closed ring carbohydrates. So what you can
Yes.
understand is periodic acid can only act on I mean it can act on multiple different substrates. In our case, if we allow it to act on proteins, let okay, let me rephrase this. The mechanism with which people use this PAS reagent is that periodic acid would act on saccharides, monosaccharides or disaccharides, any sugar molecule. And these sugar molecules have OH groups, alcohol groups. Now, upon oxidation, alcohols would turn into aldehydes and the ring would open. Then comes the shift base, which sits on there and gives you the color. This is how it works. That is why periodic acid doesn't is not just used for staining glycoproteins, but also for anything that can store glycogen or any glyco-related moiety, anything with sugars. Now, you can think of this question and maybe try to answer next week. We have specific functional groups like in serine and threonine, which also have an alcohol group. Okay. Do you guys remember the structure of serine and threonine? So, in serine, what would happen is in the R group, you'll directly have OH. Whereas in a threonine, you will have in a serine, you'll have a C and a OH. In a threonine, you'll have a C and another C, which is linked to another C. So this middle C would have an OH. Okay. So this is also now having an alcohol group. So this is like a thought exercise now for you. You can think and you can let me know if this alcohol would also be oxidized, and if it is getting oxidized, will Schiff's reagent be able to stain it? Just think and let me know, maybe in the next class. Okay. The next question is in a Western blot experiment, the actin protein is probed with a mouse monoclonal primary antibody. The secondary HRP conjugated protein should be, and four options are given here: goat anti-rabbit HRP, horse anti-mouse HRP, rabbit anti-goat HRP, rabbit anti-horse HRP. So, which of these options do you think is correct? We'll start with the basic idea of the Western blot experiment itself. So, can anyone explain how a Western blot experiment starts? Okay, if not that, does anyone know why you would want to do a Western blot experiment?
So protein detection.
Huh? That you can do with normal STS page, right? Is there something specific about Western blot?
By using antibodies.
And what would antibodies do?
They target particular proteins, so it provides high specificity.
Right. That's the answer. That's the reason why we use Western blot. So in Western blot, you aim to visualize proteins having specific peptide sequences. Like for example, for his tag, we have specific antibodies that would target the his tag to identify his tag proteins and so on. So in this process, we would first run an SDS page, then transfer the contents of SDS page to a membrane. Once we transfer it, we incubate that membrane with our tag of interest or our protein sequence of interest. If you have an antibody that is raised to detect his tag, then that can be used. So we put that particular antibody that is called the primary antibody. Then we would put another antibody that would target the primary antibody. This is called as the secondary antibody, and we would detect based on the secondary antibody. So this is the entire uh, flow of this experiment. So the question here is, if the actin protein is probed with mouse monoclonal primary antibody, the secondary HRP conjugated protein should be what? So what one needs to understand here is that independent of what uh, your primary antibody is targeting, it can be a his tag, it can be a GST tag, that doesn't matter. As long as your antibody is raised in a particular organism, like in this case, they say it is mouse. If it is raised in mouse, then the secondary antibody should be raised in an organism that is anti-mouse. That is the logic here. Which is why among the four options given here, horse anti-mouse HRP is the right answer. HRP is nothing but horseradish peroxidase, which is an enzyme that is linked to the antibody. And if you give it a right colorimetric substrate, you will be able to visualize your protein. Okay. The last question is, a dark brown precipitate is formed when a blot is developed with, and four different options are given here. So here the option diaminobenzene and nickel chloride is the one that forms dark brown precipitate. Other than nickel chloride, cobalt chloride can also be used for the same purpose, where you will see a difference in the color. So with this we come to an end for this week's lecture. If you have any doubts, You can ask me. Otherwise, we'll end the call.
So can you explain the 15 this question again?
Okay, so this question is about immunostaining, histochemistry. So what they ask you is, in which of these blot development methods do you see a dark brown precipitate? So what is observed is when you add diaminobenzene, benzene or benzamine, I'm not sure about the full form of this particular molecule. So when this interacts with tissues having particular molecules, what would happen is this would bind to them. Then you can add different dimetal cations. It can be nickel, copper, or cobalt, depending on which the color change would happen. When you add nickel, it turns into brown. And there is a paper referenced to this particular technique of immunostaining. I'll share that. Uh, this is how much I know about this particular methodology. If you have any specific questions, you can ask me. I'll try to find the answer and I'll discuss that. Okay. If there are no more questions, I'll end the call here. Thank you.
Thank you, sir.
Thank you.
Thank you.
ஒரே நிமிஷமா நான் வெளில சாப்பிட்டுலமா இத பத்தி உனக்கு எப்படி தெரியுது? பின்ன எதுக்கு என்ன கல்யாணம் பண்ணிட்ட? என்னம்மா என்ன ஆச்சு? வீட்டுல சாப்பிடவே மாட்டியா?